Hi, my name is Kiala Pelekai. I am a master's graduate student in Dr. Jessica Miller's lab here at Hatfield. I'm also a member of the Coastal Oregon Marine Experiment Station and NOAA's Living Marine Resources Cooperative Science Center. I study Pacific lamprey. Pacific lamprey are native to northern regions of the Pacific Ocean and the freshwater rivers that lead to it. They have an anadromous life cycle, which means that they are born in freshwater, migrate to the ocean to feed, then migrate back to freshwater to reproduce. Pacific lamprey are parasitic in the marine environment, where they feed on the blood and flesh of other fish species. They are also semiparous, which means that they die after spawning, so their life history is very similar to the salmon of the Pacific Northwest. Pacific lamprey play ecologically important roles in freshwater systems. The returning adults are rich in fats and other nutrients, which they transport from the ocean to freshwater rivers and streams. And as you can see by these images, they are preyed upon by numerous terrestrial and aquatic organisms. Unfortunately, the abundance and distribution of Pacific lamprey have declined throughout their native range. In the US, they are listed as a species of concern. More information is needed on the biology and ecology of this species to better inform conservation and management efforts. That's where I come in. The goal of my research here at Hatfield is to use Pacific lamprey anatomical structures to learn more about their biology and ecology. Specifically, I study Pacific lamprey eye lenses and statoliths. The eye lenses of Pacific lamprey are very similar to the ones that humans have. They continuously grow layers, similar to the layers of an onion. My work involves peeling these layers and analyzing them for carbon and nitrogen isotopes, which are ecological tracers that can be used to infer feeding and geographic movement trends. I am studying Pacific lamprey eye lenses to see if I could use these layers to reconstruct their lifelong feeding patterns. The statolith is a small calcified structure found within the inner ear of lamprey. It serves the same purpose as otoconia do in humans, which is balance and orientation. Similar structures found in other fish species can be used to determine the age of fish by counting the growth rings, which are similar to the rings found in trees. I'm studying statoliths to see if they can be used to determine the age of individual lamprey. Thank you for coming to my talk. Bye.